Good afternoon, everyone. Thank you for joining us for today's events. My name is Kristen Mercier. I'm the Assistant Director of Advancement Operations for the Department of Chemistry. Before we hear from our speakers today, I'm going to walk us through a couple housekeeping items. Firstly, the chat is available for your comments throughout the event, so feel free to introduce yourself, share where you're joining from, maybe some words of encouragement for our student speakers. You can also use that to let us know if you need technical assistance. We should have some time for audience questions following each student presentation. So you can also use the chat for those, but for those, we ask that you send those directly to Professor Scott Silverman, who's going to moderate the Q&A. So you can do that by double clicking on his name in the participants window to send a chat message directly to him. And again, if you have any trouble with that, just put your question in the chat and we can help you with that. So now it's my pleasure to introduce Professor Scott Silverman. He's been on our faculty since 2000 as a professor of chemistry and also serves as our associate head of budget and operations. He's going to briefly talk about kind of the importance of undergraduate research in our department to provide some context for the presentations we're about to hear.
Thanks, Kristen. Thanks, Amanda, for organizing this wonderful student research showcase. I'm going to be very brief in my comments because the feature here is really the students themselves. Undergraduate research is just really important for the laboratories and for the students themselves. It's important for the laboratories here at Illinois because these students truly contribute a lot to the labs themselves. They aid graduate students and postdocs with their research, and in many cases, they have research projects of their own, which then is valuable for the students themselves as they're figuring out their own career paths, figuring out what they want to do with their degrees from Illinois. And I think you'll see today 4 really great examples of the students at several different levels, sophomore, junior, and senior, of their research accomplishments and a little bit about them themselves from their own words. So without further ado, I'll introduce our first of the 4 speakers. This is Sungwook Kim, who's working with my colleague Hee-Sun Han, and Sungwook is a rising senior in chemistry. Sungwook, the floor is yours.
Hello, my name is Sungwook Kim, having Peter B. Scholarship for undergrad research from Chemistry Department in Hana. Today, I would like to discuss about my research project, Droplet-based amplification and detection of circular viruses. First, I would like to talk about the backgrounds and the goals for this experiment. The circular RNA is a specific single-stranded RNA that forms a closed continuous loop through the covalent bonding. And this circular RNA has been linked potential list of RNA developing for being a cancer cell or a tumor. So our goal for this experiment is to quantify the circular RNA as function of disease stage and investigate the potential use for early cancer diagnosis and disease prognosis. During the experiment, we have used method, the double emulsion droplet made by microfluidic, PCR for amplifying the RNA and DNA, and several other detection methods. So double emulsion droplet, which is used for this experiment, is consistent with 3 layers, which is water, oil, and water droplet. And the inner aqueous consisted with the DNA templates, PCR reagents, and buffer and water. And for the oil phase, it is consistent with the HFE oil. And for the outer aqueous, it was prepared with the buffers, waters, and surfactant. The reason that we have used the double emulsion droplet is based on our group's previous research, which introduced for having an advantage that the double emulsion droplet is robust after the PCR reaction. So it provides the constant amplification factor than other droplet methods. For this method, it is precaution that these drops have to be monodispersed. Therefore, for the each experiments, we have used the same flow rate to make the constant droplet size. To make the double emulsion droplet, the microfluidic method was used. This is the scheme of the microfluidic channel, which is made by PDMS. The left side of the channel is injection part, which is hydrophobic as we set the fluids to flow left to right. The outer aqueous, oil, and inner aqueous are injected in order to left to right. The right side of the PDMS is the outlet and the collecting part of the droplets, which is hydrophilic treated by plasma cleaning. So there are two junctions which creates the double emulsion droplet. At the first junction, the oil covers the inner aqueous and forms the single emulsion. And for the second junction, the outer aqueous covers the single emulsion droplet, then forms the double emulsion droplet. In general, PCR are known as COVID tests but actually PCR, the polymerase chain reaction, is the method which copies the specific DNA sequences and helps amplifying the DNA. In this experiment, due to low concentration and small size of the circular RNA, having only one PCR, results the low abundance for detecting. Therefore, the PCR was held twice. In this experiment, reverse transcript digital droplet PCR was used for pre-amplification, and the digital droplet PCR was used for the amplification. So now I'm just going to introduce briefly about some steps. First, we encapsulate the double emulsion droplet and having the pre-amplification with the RT-ddPCR. As I introduced from the previous slide, the ddPCR will be held for amplification. But before that, there's a step for breaking droplets and encapsulating the second double emulsion droplet. The first droplet has to be broken for the second droplet encapsulations. Since the pre-amplified DNA sample solutions from the first droplet inner aqueous has to be used for the second droplet inner aqueous, and the oil should not be contained. Before breaking the droplet, the calculated outer aqueous was extracted because when the droplets are broken, the inner aqueous are mixed with the outer aqueous. Then the this presentation, the bottom part shows the calculations of the extractions to set the dilution factor, and the dilution factors are important to have a precise experiment. Coming back to the workflow, after the droplet breakdown and second encapsulations of the double emulsion droplet, and then we're going to have a ddPCR for the amplification. Afterwards, the viruses of the DNAs was detected by three methods: gel electrophoresis, flow cytometry, and the fluorescent microscope imaging. The gel electrophoresis is a method which distinguishes the size of the DNA by using an agarose gel and electrode. By using this method, it helps to confirm the expected sequence of the DNA. The fluorescence microscope imaging helps to know whether the DNA has been sequenced by using the fluorescent and checking the size of the droplets. Checking the size of the double emulsion droplet is important since the double emulsion droplet has to be monodispersed and same. For the experiment, triplicate of the droplet has been collected and the size of the droplet resulted similar for the same flow rates. Now, I want to introduce about the flow cytometry. The flow cytometry was used to detect and count the numbers of the negative and positive signals of the droplets. These two graphs show the results of the negative control sample and diluted DNA template droplet sample. The x-axis VCA is a signal of the fluorescence, which provides different signal for positive signal of the DNA and negative signals. Therefore, by comparing two graphs, the DNA templates samples are detected. In the left side of the negative control, it shows a one big peak, which is only a negative signal. In the right side, it has two peaks. One big peak is a negative signal, and the right side of the peak is the positive signal. And by comparing these two graphs, we could know that there's a positive signal. And by using the software for flow cytometry, they give a percentage proportion of the positive signals. So for the ddPCR, we're expecting to have same DNA amplification factor for different DNA templates. So we had to experiment using the three different dilutions DNA template solutions. So from the detection method has provided the same size of the droplet and the same portions of the different portions of the positive droplets. By using this data, the Poisson distribution was used to calculate the concentrations of the amplified DNA samples from the PCR. By utilizing the Poisson distribution, amplification and DNA concentration amplification factor was calculated. As a result, the different concentrations of the DNA template solutions have shown similar amplification factor, which means that ddPCR measurements are quantitative and reproducible. So for the conclusion, the different concentration and triplicate solutions show the same size, similar size of the droplet and similar amplification numbers. So we can say that ddPCR measurements are quantitative and reproducible. And for our future plan, we're going to use the unknown concentration circular viruses from the patient plasma sample, and will be quantified and correlated to the disease stage and gene expressions. I want to briefly introduce myself. I got interested to chemistry since middle school because I thought everything in the world was related to the chemistry sciences. So nowadays, I'm specifically interested into the organic chemistry and the biochemistry, since developing the health sciences could make us live longer and healthier. So I'm planning to go into the grad school for these fields. Lastly, I just want to acknowledge to Dr. Heesun Han and Han group members supporting me to learning about the researches, and thank for the Peter B. undergrad scholarship donors for supporting me. And this was end of my presentations. Thank you for listening.
All right, thank you so much, Sungwook. Really nice progress on your research, very interesting. As Amanda is saying in the chat window that I hope everybody can see, if you have questions, you can send them along in the chat, and I will be happy to pass them along to our student speakers for the entire hour today. So just to get started, I have a question of my own I can ask you. In your microfluidic setup, can you easily change the droplet size? Is that something that you can tune?
Okay, for the macros, going back, we can just change the size by using the different flow rates.
Okay. And is that something that's valuable to do?
For me, I think it depends on the reagents that we use. But since we wanted the constant amplification, the size of the droplets matters a lot. So we're just using the same flow rates. That's the reason that we're just having same flow rates for whole experiment.
Mm-hmm. Makes sense. So one question here is from the chat here that I can see. In order to make the droplets, have you varied the concentrations of reagents and buffers? Does that composition of the droplets impact your outcomes of your experiments?
Yeah, having so by using the buffers and like kind of things, the DNA templates and the concentrations, it differs the results for having the bright drops, but for the result, it showed the same amplification factors. Like even though we have we have 1 or 2, it's gonna multiply the same factors. Like for example, if you're just having like amplification factor 2, and we're PCRing 1 DNA sample, it's gonna give a result for having 2 copies for the DNA samples. And if you're having 2 copies, you're doing the 2 DNA template for PCR, we're gonna have 4 DNA copies for the results. So the concentrations doesn't matter for just amplifying, I guess, for our results.
Okay. All right. So I think in the interest of time here, because we have about 15 minutes or so per student, we'll move on to our second of our 4 speakers. This is Aidan Lindsay, who's working with my junior colleague, Nicholas Jackson. And Aiden, you can go ahead and show your screen.
Right. So my name is Aiden Lindsay. I am a chemistry major in the class of 2025, and I've been doing research in the Jackson Group this summer. And I'm very fortunate to be supported by the Thomas Rebeck and Susan Morisato Scholarship in Chemical Data Science. So the title of my project that I'll be presenting with you is Applying Machine Learning to Doped Organic Systems. So the revolution of electronics in the last 100 years has had a profound impact on many aspects of our society. Semiconductor technology in particular has touched a variety of important industries to our everyday life. For example, the photovoltaic, lighting, computing, healthcare, telecommunication, and automotive industries. And this is in no small part due to doping, which has been able to greatly increase the electrical conductivity of these semiconductors. Recently, organic semiconductors, or OSCs, have emerged as more flexible and cheaper alternatives to the traditional inorganic semiconductors that are currently widely used now. But they require about 5 orders of magnitude more dopant to be conductive. And this can lead to significant morphological, meaning structural, disorder in the organic semiconductor, which can further inhibit conductivity. So the key question of my project is, how does the morphology of the organic semiconductor affect its electrical conductivity? As a better understanding of this structure-function relationship will allow us to engineer more highly conductive OSCs in the future. And so to this end, my project is about developing a machine learning model to model the relationship between morphology and the electronic couplings of organic semiconductors and dopants. So firstly, what is doping? So doping is the introduction of a small amount of impurity into a chemical substance, in this case, the organic semiconductor, to increase charge carriers in the material and hence increase its conductivity. So here, this figure here on the left is an organic semiconductor. It's characterized by these sort of aggregated regions and more amorphous regions. And when you introduce a dopant into the structure, a reaction occurs in which the dopant gains a negative charge, and then we have positively charged holes in the organic semiconductor. And these positively charged holes are what carry electrons through the material and make it conductive. So in my project, I am using P3HT as a classic organic semiconductor and F4TCnQ as the dopant. And so again, the goal of my project is to see how does the structural relationship between P3HT and F4TCnQ affect the generation of charge carriers in the organic semiconductor. So the previous slide had a pretty simplified view of doping, but when we look at doping, we're looking at 2 main doping mechanisms. The first is integer charge transfer. So in this mechanism, the highest occupied molecular orbital, or HOMO, of the OSC donates an electron to the lowest unoccupied molecular orbital, or LUMO, of the dopant. And this involves a complete transfer of charge. And so this is typically characterized by very little overlap between the HOMO of the OSC and the LUMO of the dopant. And it's considered, of the 2 doping mechanisms that we're looking at, the most desirable doping mechanism because there is that full transfer of charge. The second doping mechanism is called the charge transfer complex. So in this mechanism, the highest occupied molecular orbital of the organic semiconductor and the lowest unoccupied molecular orbital of the dopant hybridize, and they form this charge transfer complex in which a partial charge transfer occurs. So it's a little bit less desirable than integer charge transfer. And in the case of P3HT and F4TCnQ, this often occurs through ring-ring stacking. So the rings of P3HT and the ring of F4TCnQ undergo pi-pi stacking, and this charge transfer complex forms. So in order to investigate the prevalence of these doping mechanisms in the P3HT F4TCnQ mixture and look at its relationship to morphology, its morphological relationship to the conductivity of the organic semiconductor, I ran a molecular dynamics simulation to see how these molecules interact on a molecular scale. So this was run for a total of 20 nanoseconds at 1 femtosecond time steps. So molecular dynamics essentially solves Newton's equations for these molecules in a simulation box to see how they interact with each other. And so you can see this on the left is the final frame by molecular dynamics simulation. And it's a bit hard for me to just look at this image and gain any structural insight into what maybe common structural motifs are happening. So to that end, I need to perform some structural analysis to investigate the dopant effect on the morphology of the organic semiconductor. So I computed some radial distribution functions. And what these radial distribution functions, or RDFs, are is they essentially describe the clustering of molecular pairs with respect to an ideal gas, which is normalized to 1. So essentially, like peaks above 1 means it's more clustered than the ideal gas, dips below 1 means less clustered. So I was able to draw several conclusions from these RDFs. So first, in this upper left RDF, which is between P3HT, our organic semiconductor, monomer pairs, we can see that in the region between 5 and 7 angstroms, the P3HT oligomer chains between 5 and 7 angstroms is like the nearest neighbor distance between those chains. In this upper right RDF, which is between our dopant molecules, we can see this large peak that occurs around 7.5 angstroms. And what this indicates is that there's significant dopant clustering within our system. And so this is significant because dopant clustering can actually inhibit charge carrier generation and hence inhibit the conductivity, since the dopant anions are clustered together and not really generating as many positively charged holes in the organic semiconductor as they could be. And the amplitude of this peak essentially just correlates to roughly clusters of 3 F4TCQ molecules. And then finally, these bottom 2 radial distribution functions are between our OSC and our dopant. The one on the left includes the full structure, while the one on the right only includes the ring of P3HT. And we can see when we only include the ring, there's this large peak that shows up. And so what this indicates is that ring-ring stacking between P3HT and F4TCQ is pretty prevalent in the simulation. And we know that ring-ring stacking typically correlates to charge transfer complex formation. So the next step for me was to start to establish my machine learning data set. So in order to do this, I wrote a script to extract closely located pairs of P3HT monomers and F4TCQ molecules. And I ended up extracting about 2.2 million different pairs from my molecular dynamics simulation. And while unfortunately, I cannot show you all 2.2 million different pairs, I can show you 2 representative pairs that very clearly represent the 2 doping mechanisms that we're looking at. So the first is a clear case of ring-ring stacking. So we can see this is the F4TCQ dopant. This is a P3HT monomer. We can see that these rings are very clearly stacking on each other. They only have a 4 angstrom separation. They're very close to each other. And my postdoc, Chuni, did some quantum chemistry calculations on these on this pair, and we can see that the highest occupied molecular orbital of the organic semiconductor and the lowest unoccupied molecular orbital of the dopant overlap a lot. So if you were to superimpose these 2 images, there would be a lot of overlap there. And we know that leads to a charge transfer complex formation. On the other hand, another pair that we extracted showed the other mechanism, which is integer charge transfer. So there was a much larger separation between the organic semiconductor and the dopant molecule. And you can see if you were to superimpose the HOMO and the LUMO, there would not be much overlap. So we know that a full transfer of charge would occur in this case. So the next steps of my project are to take my current data set of 2.2 million pairs and pare it down to about 50,000 representative pairs. Is when you have 2 million different pairs, there's going to be a lot of structural redundancy there. So to do this, I will be conducting principal component analysis. An example of a PCA plot is right here on the right. And this will allow me to identify shared characteristics via clustering in my data points. That will allow me to reduce my data set size into a more representative 50,000. And then after that, the overall goal of our project will be to use machine learning on a reduced data set to create a model that can predict the electronic couplings from the dopant OSC structure. So we will take our 50,000 pairs and essentially feed it into DFT calculations, and that will be our data set that we will do machine learning on. And of course, this model will provide much needed insight into the relationship between conductivity and morphology in organic semiconductors. So a little bit about me. UIUC's very well-reputed chemistry program brought me here from Kansas. I became interested in chemistry when I participated in high school level competitions like the US National Chemistry Olympiad. The highlight of my research experience was working with my wonderful postdoc mentor, Dr. Chuni Wang, and I aspire to a research career in computational material science. So thank you to the Jackson Group for welcoming me for the summer. Thank you to Thomas Remek and Susan Morisato for sponsoring me, and to everyone else for listening to my talk.
All right. Thank you so much, Aidan. That's really wonderful. Nice. And I see that Thomas Remek is here as a participant in our presentations. Thank you so much for supporting this kind of research. Very,
Yes,
very
thank you
valuable.
for
You can
coming.
see the impact on the students. If anybody has any questions, as Amanda has noted, you can put them into the chat. Aidan and I can get started with these MD simulations. I imagine you have so many outcomes that the results are essentially reproducible, right? That you have a lot of redundancy, that you get the same outcome from different runs.
Yes, there is a lot of redundancy, and that's another reason that we want to reduce the size of our data set. Right now, we've only done like one actual molecular dynamics run, but we are planning on doing multiple other runs with different initial configurations to sample more equilibrium configurations. Yes.
Yeah, makes sense. Is there a simple way to think about how closely the HOMO and LUMO have to be matched in order to have the charge transfer be effective?
So the pictures that I had in my presentation is very qualitative in nature. So I guess there isn't really like a big cutoff of like, you have this percentage of overlap that will lead to integer charge transfer or charge transfer complex. But we more wanted to show very clearly that like there was a lot of overlap here, there was not a lot of overlap here. So one will predominate over the other. But yeah, it's a very qualitative picture. It's not really quantitative. So yeah, it's hard to, I guess, put like a ratio of overlap to that.
Right.
Yeah.
Is that something that would be important as a longer term objective to have kind of quantitative numbers on that, or is that something?
Perhaps, yes, we are starting to think about ways to quantitatively characterize charge transfer complex formation and versus integer charge transfer. So yes, that is something that we're looking at. Right now, we really just have a qualitative picture, but we would like to have a quantitative picture there.
And Thomas Remek has a question for you, which is, do you think that this is headed towards a publication?
Perhaps. I think we are still very early in the project. We haven't implemented our machine learning onto our data set right now. We're just trying to get like the data set as good as possible before we actually implement our machine learning model, but perhaps in the somewhat far future. But yeah.
Okay. All right. Well, thank you so much for the wonderful presentation. All right.
Thank you.
Okay. And our third of our four presenters today is Maria Massa, who's a rising junior in my own lab. And Maria's research is on DNA enzymes. She's going to give that presentation now. Go for it, Maria.
Hello, everyone. My name is Maria Massa. I'm a rising junior in chemistry. I work in the Silverman lab. And today I'm going to talk to you about DNA enzyme catalyzed site-specific anisylation of DNA nucleobases. So in my lab, we work on developing biopolymer catalysts. So there are three main types of biopolymer catalysts. There's DNA enzymes, which consist of DNA. They're polymers of deoxyribonucleotides. And then similarly, there's ribozymes and enzymes, which are polymers of ribonucleotides and amino acids, respectively. So I'm going to talk mostly today about DNA enzymes, since that's what my lab works with. And specifically, we work with identifying DNA enzymes using an in vitro selection method. So this is a diagram that kind of depicts the in vitro selection method that we use. Let me get the laser. Okay, so in this, we start off with 10 to the 14 sequences of DNA. And so these 10 to the 14 sequences, they have a 40 nucleotide region, which is random. And so this represents our pool, initial pool of DNA, and the gray dots represent the uncatalytic sequences, but the red dots represent the sequences that catalyze the desired reaction. So we challenge this pool through an enrichment step to perform this desired reaction. And after this enrichment step, the ratio of red dots increases versus the ratio of versus gray dots. And so after that, we amplify this enriched pool using PCR. And so we iterate these two steps over and over again until we get the desired catalytic activity in our pool. And so you might be asking yourself, why would you want to identify these DNA enzymes? So one reason would be to just expand the scope of catalyzed DNA reactions. And then another one would be to develop these enzymatic tools. So DNA enzymes could be used as a real world application for nucleobase modification, specifically DNA and RNA labeling for biophysical and biochemical studies. So there's a couple ways to do this right now. For example, in this paper, they wanted to study the folding of this ribozyme. And so they did this oligonucleotide hybridization. And so that just means that they put in these loops of DNA, and then they synthesized another piece of DNA to base pair to it so that they could put these little tags in order to study the folding. This works. It is a bit invasive and it's kind of bulky. It doesn't work great. And then there's also bottom-up splint ligation, where you can just surgically put in these tags into, for example, a ribozyme, but that would require synthesizing different pieces of DNA and then splint ligating several of them together with the modified tags on it. And this also is possible, but is a lot of work and very time consuming. So a better approach would be something more top down, where you could already have the existing, for example, ribozyme, and then site specifically modify it. So that is something that we could do with DNA enzymes. A top-down approach would be much more convenient. So specifically in my research, I work with trying to identify and characterize DNA enzymes for nucleobase N-acylation. So this is kind of a zoomed in picture of the dot picture I showed you earlier. So this consists of a DNA substrate, which I call the left-hand substrate, and that has 3 overhanging nucleotides. And so the exocyclic amine from one of these nucleotides can attack in another piece of DNA that has an aryl ester acyl donor. And so in my project specifically, this aryl ester acyl donor is a tetrafluorophenol acyl donor. And we, in my project, we're examining the C, G, and A exocyclic amines as these like overhanging, for these overhanging nucleobases as a nucleophile. So yeah. So in a previous selection, one of the former undergraduate researchers in my lab, Caroline Kaminsky, she did a project where she just looked at an acylation of the C nucleobases as the overhang. And she found 5 DNA enzymes from this project. And one of those DNA enzymes, AJB210, gave robust yield with both DNA and RNA. And this was very encouraging for me, as it suggested that some of my DNA enzymes could also do this. So yeah. And so me and Morgan, another undergraduate researcher that was in my lab, we developed these selection pools over many months. And so this picture depicts the selection round by activity progression inside the pools. So these pools is kind of like depicted as a 3x3 grid. So here is the different overhanging nucleobases that were in the substrate. And then here is the pH that each pool was that was in each pool. So for example, the MK2 selection pool had the C overhanging nucleobases, and the reaction was conducted at pH 6.5. And so from these pools, 5 of the different pools gave catalytic sequences, and overall we were able to get 21 different DNA enzymes from this project. And so after we identified these DNA enzymes, we wanted to characterize them. So we did a kinetic analysis of the DNA enzymes. So here is a kinetic plot that we got. And so this depicts the kinetics of the AJB210 DNA enzyme. And so you can see it's a function of time, and then the yield goes on as time progresses. And so some of these have half-lives short as 1.5 hours, which is pretty reasonable on the human's time scale. We also did RNA substrate tolerance. So if the left-hand substrate was RNA instead of DNA, and 9 of 26 worked with RNA. And we also did reaction site determination. So for reaction site determination, there were 2 different nucleophiles that could have participated in the acylation DNA enzyme reaction. So one of them is the exocyclic amine. And so if that was the nucleophile, if that was acylated in the reaction, it would have formed an amide. And if the 3' hydroxyl, which is all the way at this end of the overhang, was the nucleophile in the acylation reaction, an ester would be formed. And so for the cDNA enzymes, we used a modified left-hand substrate. And so this modified left-hand substrate, instead of having a regular C nucleotide here, which is portrayed here, we used a DDC nucleotide. And so that is just, it gets rid of the 3' hydroxyl. So instead of the 3' hydroxyl here, it's just like a hydrogen. And so we used this modified substrate and we compared it to the regular parent substrate. And so we performed the reaction and we saw, as you can see on this page gel here, that the yields for the modified and the parent substrate are very similar, meaning that the 3' hydroxyl is not participating in the reaction. We also, however, for the G and the A DNA enzymes, dideoxy-G and dideoxy-A are not readily available. And so we're going to do hydrolysis assays to determine the nucleophile for those DNA enzyme reactions, as esters are more stable, esters are less stable, so they'll hydrolyze faster, and amides are more stable, so they'll hydrolyze slower. So we also wanted to determine which of these overhanging nucleotides were participating in this acylation reaction. So we did this assay where we extended this 3' end of the left-hand substrate. And then the products that we got from after we did the DNA enzyme reactions were more branched. And so on a page gel, we were able to identify which the site based on like the branching and the migration pattern on this page gel. And so as you can see here, we have C+1, C+2, and C+3, and 8 of the 9 DNA enzymes that were tested are 95% selective. And as you can see here, this one reacts at both C+2 and C+3, so it's not site selective. Additionally, for the cDNA enzymes, we did a completely different assay where 5 different products are made for each DNA enzyme. And these different products had a modified substrate where a ribonucleotide was put in the +4, which is all the way at this 3' end, like past the +3, and to the -1, which is at this end. And so after we made these 5 different products and did the DNA enzyme reaction, after we made these 5 different products, we treated them with base. And so after we treated them with base, we got these patterns on the page gel. And so these patterns were able to, we were able to identify which nucleotide was being acylated in the reaction. And so as you can see here, we identified that the reaction was happening at C+1 because of these 4 bands and the absence of this band here, and C+3 for AJB229 because of these 2 bands and the absence of these bands here. And so these match up very well, as AJB210 was determined to be reacting at C+1 in this completely different assay, and AJB229 was also determined to be reacting at C+3 in this assay. So everything matches up, which is great. So for future work, we want to complete the manuscript for publication. This project has been led by 3 different undergraduates, the 2 that I mentioned previously and myself. So we're going to be first co-authors on that publication, which is really exciting. We also want to do future selections including selections with a DNA substrate rather than an RNA substrate, and then selections with a free small molecule ACL donor. So in my project, the ACL donor is tethered, it's base paired to this part of the DNA enzyme. And so we want to do it where it is free in solution. And then we also want to do selections with both RNA, an RNA left-hand substrate, and free small molecules. So about me, I'm from Blacksburg, Virginia, and the excellent chemistry program here brought me to the University of Illinois. The highlight of my summer research was probably learning to use the DNA synthesizer because I thought it was pretty cool. And after my undergrad, I would like to attend grad school. I would like to thank Dr. Silberman and the former and current members of my my lab, along with Fred C. and Josephine C. Faulkner, for the scholarship this summer.
Thank you so much, Maria. And again, if anybody has any questions here, you can put them into the chat. And I'd like to acknowledge, I see Fred Faulkner as one of our attendees here. So thank you very much, sir, for your contributions to allowing this kind of research to happen. Very important. You can see the impact of it. So I'm going to feel awkward asking questions of my own students. So there is a question already in the chat. Dana Delot's asking if there's a possible in vivo applications of these or perhaps other kinds of DNA enzymes. Like what would have to be done to make these work in vivo?
I could envision like in vivo applications for maybe like protein modifications, or like there are some genetic diseases where like nuclear-based modification could be used, but so there I could see some possible like applications.
Right. Right now we're doing everything in vitro, right?
Yes.
Yes. And we're right on time here for our fourth of our four students to present. And so this is Aidan T. Arena Albury, who's a rising sophomore in Dana Delot's lab. And Aidan, you can go ahead and get your presentation showing.
Thank you for the introduction. Okay, so to get started, to get a better idea of the work I've done this summer, we kind of need to talk about the fundamental functionality of a bomb, like what happens upon that explosion. And so at a much more basic fundamental level, it's a bomb is really a high energy molecule going to a bunch of low energy products. And so from this reaction of all the bonds breaking in this molecule, you get heat and gas, and then also light. You know, we've all seen explosions in movies. There's light as well as sound and pressure and all of that. And so to get a better look at this fundamental functionality, we created a model bomb that was just a single crystal of this explosive called HMX, which is one of the most powerful commonly used explosives. And we created this at a scale of like, each crystal was maybe around 50 to 200, something like that, micrometers in diameter. And the Dot group invented a way to see inside of a bomb, basically, essentially, at this simple level and capture its first events within the first 400 or so nanoseconds. And the question we're asking with this model bomb is, where does the reaction start, depending on a number of different factors in the crystal itself, as well as other things. And so you may be asking, how did we see inside of this bomb? So what we did is we had individual wells filled with a clear polymer named PDMS, and we would suspend one single tiny crystal in this well, and we use a laser pulse to punch out an aluminum flyer that would travel at super high speeds of like 3.6 kilometers per second, and it would impact this sample, and the shock from that impact would create this explosion that from underneath in this model we can view the thermal emission from that reaction using a microscope and an extremely high-speed camera. And so the impact of this is to get a more fundamental understanding of how bombs work in the moments after impact. Like I said, nanoseconds after, it's a really interesting area to be looking at, because you really get to see the stages of the explosion. And we're looking at with any sort of big problem, you tackle it small part by small part until you get a better understanding of how to tackle the whole thing. So we're just looking at this small little guy in the big scheme of things, this one crystal, and we're looking at how do differences in that crystal affect it. And then we can hopefully apply that knowledge to larger things like a plastic bonded explosive, which is closer to what a bomb is. And then also, as you can imagine, it's good to have this model to see with high spatial and temporal resolution what is happening when really anything impacts something else. And so from this, the results we got, we really primarily looked at two different types of crystals, and one was a perfect crystal with no defects in it, and the other one was a defective one. And the defective one, as you can see, has it can have cracks in it, it can have holes in it, and the perfect one, you don't see any blemishes like that, and this you can see visually. And for comparison, off to the bottom left, there's the flyer plate. It's not this small thing. It is pretty big compared to the sample that is hitting this sample. And what we found is in the perfect crystal, the start of this explosion really primarily happened at like corners and edges of this crystal, more where the crystal is interfacing with the clear polymer it is suspended in. And in the defective crystal, we found that in those defects, in those like little cracks, is where the hot spot in this explosion tended to propagate. And the hot spot, this whole thing, the crystal image is overlaid over this like red image of this explosion right now. The whole thing is extremely hot, but the hot spots are just hotter than everything else, thousands of degrees Kelvin. And so it's where it's specifically like, as you can tell from the name, it's a hot spot. And so the next level of this would be to take a look at multiple crystals packed together. So in this sample pack to the right, that's about eight or so crystals just packed together. And we still want to look at this 2D cross section that we're looking at through the microscope of this sort of almost like 2D plane of crystals. And we want to see how great is the influence of each factor, the cracks in each crystal and holes in each crystal versus the crystal to crystal interface when there's more than one crystal, or the crystal to polymer interface. And maybe seeing if we change the interface, the thing that the HMX is in, rather than having the PDMS polymer, if we replace it with something else, how might the PDMS react with that? Would be something else we could take a look at. And a little bit about me is I grew up in Illinois, so I always heard of the U of I. I always heard they had a great science program with an amazing chemistry department. And what interested me particularly about the science and letters major is that I had more freedom to take other science classes. So last year, I still took my basic chemistry classes, but I also took a forensics class and a class on pandemics, and I found that really interesting. And I've been interested in chemistry since my sophomore year of high school, where I took an honors chemistry class, and I had I had a teacher who cared so much about chemistry, it was almost infectious. And so I decided I'd take AP chemistry. I joined the Science Olympiad team where I participated in the chemistry lab event, and we were first in the region for chemistry lab, my team, and we actually went to state here at U of I last year. And now that I'm in college, this summer I've been able to have the amazing privilege of working with the Droplet group. And obviously, as you can imagine, this equipment with lasers and very delicate explosives, it's very expensive stuff. So it's been a little bit high stakes, but very exciting to work with. And in the future, the current plan is grad school, where I think I'd like to focus on some applications of computer science and chemistry, such as artificial intelligence and machine learning, and see how those interact with chemistry and what we can learn from that. And I just really quickly want to acknowledge my mentor, Shiva, who has worked with me throughout all of this and has really been so helpful. And then Dr. Dlap, who is here, who's the professor at the head of all this, and just does amazing work, and all my other coworkers in the Dlap group. And I'd also like to thank Milan Mrkic for his generous scholarship that has allowed me to spend the summer doing this awesome research.
All right, thank you so much, Aiden. And Milan Mrkic was himself an undergraduate student in our department, as it turns out. So really great presentation. We already do have one question in the chat for you. One of the participants would like to know what the chemical reaction is that actually causes the bomb to your crystals to detonate.
Yeah, so upon this impact, this shock, all of the bonds that are in this high energy molecule, because it's such a big molecule, it kind of just breaks and it creates these gas products and a huge release of energy. Going from a high energy molecule to low energy products, that energy has to go somewhere. It releases a lot of heat and a lot of pressure from the gas molecules that are coming off of this higher energy molecule. You could have carbon dioxide, oxygen gas, and nitrogen gas, things like that. And that's what creates this explosion.
I see. How well do your results scale from you said more like micrometer scale of your crystals to macroscopic? Because when I think of bombs, I think of macroscopic.
Right.
And so how well do your results scale?
Well, right now, I don't really know. I'm more taking a look at the smaller level. We have had previous workers in the lab look at slightly bigger level, like the packed 2D crystals that I was talking about, but really stacking those 2D planes up into a plastic bonded explosive is where it gets more to that bomb level, and that is something we will look into in the future. But right now, it's more of just a fundamental what is happening with this one single particle of this HMX.
Have to start somewhere, right?
Yeah.
And when you have these hot spots, can you give us a sense of how much hotter they are than the surrounding regions? Is it like a few percent difference? Is it 20-fold different?
So I think they're somewhere around like 4,000 Kelvin, if I remember correctly. And they can be hundreds to even like 1,000 or 2 Kelvin hotter than the rest of the reaction, I believe.
Okay, so they really are a lot hotter than the region nearby. Okay.
Yeah.
Okay. And we have a few minutes left if there's questions for any of our 4 presenters, if anybody had something in mind and would like to ask it now. And if not, I think we can draw our student research showcase to a close. We're pretty much right on time. again, thanks to everybody. First of all, thanks to our 4 student presenters who are doing such wonderful research and took the time to make your nice presentations. I think we all appreciate how much effort it takes not only to do the research, but then to prepare it for others to hear about what you're doing. It's a slightly different skill set. And thanks to all the participants for taking your time to listen to these great students.